This video was made for any student who missed the osmosis lab. And so on the front page, you have a bunch of terms to define: solute, solvent, hypotonic, isotonic, and hypertonic. So define those from your notes. And what we're going to look at now is how to do the lab itself. So we're going to start with observing osmosis in an egg. And so the first thing to do is to write your procedure. So when you watch the video of the procedure, you can write that down. So basically what we're doing is taking eggs that I've already taken the shell off of, and then you're going to weigh the eggs, put them in beakers, and in one of the beaker you put corn syrup, and in the other beaker you put distilled water. So I want you to make a data table next. And so your data table should look something like this. I want your initial or first weight here, and your final weight here after a day, and then the difference between them you can put in here. And so this is going to be the weight of the eggs, and you want to put one that's in corn syrup. So egg in corn syrup, and you want to have one with an egg in distilled water. And maybe you can use a plus for the one that got bigger and a minus for the one down here that got smaller. This I didn't write very well. It's corn syrup. Okay. So circle the type of solvent below that the corn syrup will be in this lab. So you're going to have a beaker, and you're going to put an egg into it after you weigh it. And then you're going to pour corn syrup on top of it. And so the corn syrup has just a ton of dissolved stuff in it, and all that dissolved stuff is sugar. And so the word for having more is hyper, like hyperactive. That's a kid with a lot of energy. Hypertonic means you have a ton of dissolved stuff. So you have a little bit of dissolved stuff in the egg, but you have a ton of dissolved stuff in this corn syrup. And so that means it's a hypertonic solution. So what do you think will happen to the egg when it's placed in the corn syrup? So that's your prediction, and your hint is that water goes from a high concentration to a low concentration. So if you look out here, you have so much sugar in here that there's hardly any room for water at all. And so the water goes from a high concentration to a low concentration. And so I want to know, does that egg shrink or swell? And so that's what you're going to write here. And then circle the type of solvent below that the distilled water will be in this lab. And so here's a beaker with distilled water in it. And you put your egg in it, and there's the distilled water. And the egg is really ugly, sorry. And anyway, it has some dissolved stuff in it. Water will, well, out here, it's pure water, so there's nothing dissolved in it. So that means there's less dissolved stuff than the egg. So the word for less then is hypo. Like hypothermia means you have less body temperature. So hypotonic means there's less dissolved stuff in it. So it means there's a lot of water there. Water goes high to low, which means it's going in. And so right here, you're going to tell me whether the egg is going to shrink or swell. And so what you're going to do now is watch a video of me doing this and then doing it again a day later. Okay, so what you just saw, what happened to the egg in corn syrup and why. And so right now you have to tell me whether the egg shrank or swelled. And then you can tell me it did this because there was a higher concentration of water wherever it was, in or out, and which way the water went. And then you want to do the same thing for the distilled water. So neither of these actually popped because there's a pretty thick membrane on the egg, and so it's not just a regular old cell membrane on it, but something a little bit, but if it were just a red blood cell or something, it could have actually broken in one of those two solutions. So what you're going to look at now is observation of osmosis in onion cells, and onions are plants. So they're a little bit different. They have a cell wall in them. So what you're going to see now is a video of me getting the procedure, doing the procedure for the egg. So you're going to take a look at that now. And what you just saw, let's see, is a picture that looks like this. And so these are your onion cells. And so the purple stuff, the pretty purple stuff, is all the cytoplasm, and those white lines that you can see there are the cell walls, and you really can't see a cell membrane there at all. So take a picture of the cells that you see and print it and label the cell structures that you see visible. You can print it out from It's Learning. I have a picture just like that on there. You're going to label the cell wall and the cytoplasm, and that's really all you can see. You might see a nucleus. If you do, then you can label that. Does the cell membrane appear to be separate from the cell wall, or does the membrane appear to be right up next to the cell wall? It should be right up next to the cell wall there. And so what can you tell me about the turgor pressure of the cells you're observing? Well, turgor pressure means water pressure, and so what you saw was a cell that looked like this. And this is your cell wall. And so it was in a hypotonic solution because around here was just water. But in here, there's lots of dissolved stuff. So water came in. So that gives you a very high turgor pressure. And just to make sure you understand all the parts, the purple, the pretty purple stuff that you saw, oops, I don't know why that went away. Here, let's do this. The pretty purple stuff that you saw was the cytoplasm. And then the stuff on the outside, oops, let's make it black, I guess. The stuff that you saw on the outside, that's the cell wall. It looked really white when you were doing it, but it shows up here better black. So here's your cell wall, and the stuff inside was the cytoplasm. Whoops, sorry. Yeah, the stuff inside was the cytoplasm, the purple stuff. Okay, so what we're going to do next is put salt in. And so you're going to see a video of how we put the salt in. Okay, and then let's look at a picture of this. So you should have seen, okay, that's it. You should have seen what happened. You should have seen something that like this. And so the black lines are the cell walls, and the white space is actually just space. And you can see all of the cell membranes have shrunk in here. So I'll draw a picture of what you just saw. So what happened to the size of the cytoplasm after the water was added? And so, oops. And so you should have seen a cell wall like this. And when you had the salt water, that means there's a bunch of dissolved stuff out here. And so if you have a bunch of dissolved stuff, that means there's not as much space for water. So this is a low concentration of water. So water went from a high concentration to a low concentration. Pretend I'm drawing, well, maybe I'll finish it off. So anyway, this is all the salt that we added. So when the water leaves, the cell shrinks, but the cell wall just stays put. So the cell membrane actually gets pulled away from the cell wall, kind of like that. And so the black line that I'm drawing now is the cell membrane. The line that's out here is the cell wall. And the cytoplasm, I probably should switch it to purple, but anyway, the cytoplasm is all the stuff that's in here. It looks a little bit brighter and a little bit more concentrated, but you can see all this white space, and that's all it is, just space. So when the cell membrane comes in like this, we call it plasmolysis. So the plasma membrane has lysed or broken away from the cell wall. What do you think happened to the cell's turgor pressure? Well, that means water pressure. So clearly the water is leaving. So the pressure has gone down. And so I want you to explain that to me with words like high concentration and low concentration. And you have finished the lab. Make sure you add your pictures and label everything and answer every question.